Bom dia, pessoal. Essa é a última reunião do nosso departamento desse ano. Né? Queria agradecer a todos pela presença o ano todo. A partir do próximo semestre, a gente vai retomar as reuniões logo depois das férias, provavelmente no meio de fevereiro. Nós retomamos essa reunião depois do Carnaval. E no próximo semestre a reunião vai acontecer às sextas-feiras. Tá? Nós estamos alternando os dias. Então as equipes que têm atividades às quintas-feiras podem retomar às quintas-feiras, porque no próximo semestre as reuniões vão ser feitas na sexta. O calendário das reuniões a gente vai divulgar até o final do ano. Bom, para a nossa última reunião, we are receiving today our guest, Professor Ellen Goode. Ellen is a professor of epidemiology at Mayo Clinic in Rochester. She will talk today about population studies of immune factors in ovarian cancer. Ellen has an extensive experience in this field and about her work with this field today. Thank you very much.
Okay. Bom dia. Bom dia. Thank you, Francisco. Thank you, everybody, for coming. It's been my pleasure to be visiting you here in Brazil, and I hope that it has been fun for you as well. I've enjoyed the trees and the food and mostly the people. Thank you. This morning I thought that I would start with sharing about where I had come from. As Francisco said, I work at the Mayo Clinic, and this is located in the north of the United States by the Great Lakes. And it's somewhere in a small town called Rochester. It's about 100,000 people living there. It's not a big city. It's not a destination. And many times people wonder, why is this very nice medical place living in the middle of nowhere? So in America, we have this salad dressing called mayonnaise, or we also call it mayo. So one of the jokes is it's for the mayonnaise. This is not true. We are in the land because of this family of doctors that are shown there. There's the father, Mayo, and two brothers, Mayo. And I'll tell you a bit about them. And how they, okay. Oh, see. Obrigado. Okay. To show you how much that we like to work at Mayo Clinic, you should understand how difficult the weather is. Okay, just as we were discussing before this morning's seminar. This is a plot, because I'm a data scientist, of the data of the average monthly temperature of this nice city, shown with the highs in red and the lows in blue, and the solid lines. You have a nice climate. In Minnesota, where I live, it's very cold in the winter and very hot in the summer. It's really quite cold. So this is my first results data slide. But then, also, that's only the averages. There are outliers. There are days, for example, when this photo was taken of me by my house, it was minus 24 Celsius in the midday. We had a very cold winter this time. So to show you the dedication of the people willing to live in these places. So why are we in Minnesota? It's because of this Dr. Mayo. He was born in England, in Manchester, in 1918. And this is the time when the Industrial Revolution was starting, and the conditions were difficult for poor people versus the wealthy people. The English author, novelist Charles Dickens wrote about this, if you've read any of those books. He was born to a family where his father was an artisan and a sea captain, but he died young. His father died when this doctor was young. He did study with physicist John Dalton, and was interested in medicine early on and learned from some of the city's infirmaries, but times were difficult. So at the age of 27, this man went to the United States and was a chemist in New York City, went to other towns around the middle of the country in this place called Indiana, as well as Buffalo, worked as a tailor, went to pursue medical degree, where it took only 4 months and cost $150 in Indiana to get a medical degree, and then to another university to get a medical degree, and then kept wanting to wander, so went to this wild land called Minnesota, where he inspected the copper mines. He worked on a newspaper, was a farmer and a veterinarian, did different things. And then the United States Civil War broke out, and Dr. Mayo thought it was a good time to settle and be able to have a stable job back in medicine. Even though the Civil War was in the South, even in the North, there were some battles. And he went to go to Rochester for this reason, to work in the Army as a battalion surgeon for the U.S. Army. And they decided they liked it okay and decided to stay. Of note, he became very famous in 1871 in all of the newspapers in the U.S. for performing a laparotomy on a very large ovarian tumor of a local woman, using some of the very crude instruments that are shown here. And it was a successful medical practice, getting some fame, writing prescriptions for things like brandy, alcohol to drink, and some tree roots to eat to heal yourself. In 1883 in our town, there was a big tornado. We live in the area similar to Kansas from the movie Wizard of Oz. It decimated the town of Rochester, and many people died. And there was also a convent of nurses in the town, and the nurses said, they weren't nurses, nuns, the convent of nuns, and the nuns said, hey, doctor, please help us make a permanent hospital. And it's really the reason of these nuns and their tornado why the hospital was established, to help these people. So it's the doctors as well as the Catholic nuns. So they established this hospital. It's called St. Mary's. And then Dr. Mayo was able to stay there. He assisted in making the hospital. He established many things. His two sons, which we call the Mayo Brothers, carried on the tradition of what was then now called the Mayo Clinic. So a few things that were established back at that time are still very, very present today. We feel it very strongly in the culture. There's a unique heritage and culture to this institution. There's another doctor, Henry Plummer, who's shown here, who started it as well with them, and he had the idea to have one medical record. So no matter which specialty, it from the very beginning was a common medical record that facilitated communication among the clinicians for the best care of the patient. The hospital and the clinic stayed connected from the beginning to facilitate patient care. There was a very strong tradition started for teamwork, as well as time with the patient to thoroughly explain. And the physicians, at this time, I think it was new, received a stable salary rather than per patient or per test that was ordered. And so they set up what was a sort of a first integrated practice, at least in the United States. And today there's over 3,000 physicians and scientists like myself who are working at the institution. And there are 2 sites that are in the south of the United States, so we can escape and go visit with our colleagues in Arizona and in Florida, as well as in the Rochester site is where we are. This is a photo of our town of Rochester, Minnesota. It's not as lovely as this town, but it's okay. And we have a hospital there. Shown at the bottom is the hospital that was founded by the Mayo Brothers and father and the nuns. And then we have another campus in the same town, which has a second hospital and then the clinical facilities. So I thought that might be interesting. The bottom line is today we have a spirit of these three, call it the three shields. So we believe that, as the Mayo Brothers said, the needs of the patient come first. So we are always thinking of the patient. Even in research, the patient need always comes first. So the clinical shield is the most important. Secondary is research. So we really have a supportive research system for studying human disease and human biology. We have some very nice core facilities that the institution created. And we also have resources such as donations and donors who provide funding that the institution invests to help the researchers. So it's a very, very nice in that respect for sure. And then also the educational shield is critical. And one of the quotes from one of the Mayos was, once you start studying medicine, you never get through with it. Obviously, we all agree with that. So I went to this strange land of Minnesota in 2005. I went there to start to study genetic epidemiology in cancer, but not ovarian cancer necessarily. I had worked in other ones. But I ended up primarily focusing on ovarian cancer because the resources and the teams were just fantastic. So as in here, these are some of our numbers from the United States. Ovary is not one of the common cancers, but it's one of the more deadly cancers in women in the United States. So I'll tell you about our biorepository for ovarian cancer research that we have established and maintain at the Mayo Clinic next. So first, we of course have the paraffin tissues from surgeries since the very beginning that are saved for clinical use and available for researchers. We, in 1990, started to collect for research purposes a sample of frozen tissue from surgeries of ovarian cancer in 1990. In the year 2000, so that's now 19 years ago, we began to collect also a blood sample from these women. And as well, we started the collection of matched population controls, so women without ovarian cancer. And nowadays we have this very good resource. So women are asked to enroll. They provide written informed consent for the tumor sample, so fresh frozen and paraffin embedded, because different technologies use different things. The blood sample provides a source of DNA as well as serum and plasma. The medical record, of course, is electronic now, but that, since the very beginning of Mayo Clinic, has been great, and the participants are very willing to participate in research because of the reputation, and we have access to medical record. All of the participants are given a risk factor questionnaire for epidemiologic factors, such as lifestyle factors and other exposures. For the cases, we have permission to contact them forever. If they don't come back, we send them a letter to see how they're doing, if they have had a recurrence. Most of the women do return to Mayo Clinic for their follow-up care after diagnosis, but sometimes they don't if they come from far away. And for some cases, we have also processed the blood in such a way that we have lymphocytes that are slowly frozen so that we can develop cell lines from the women. And for some women also, we have collected more aggressively the fimbria and tube from the surgeries. So all in all, we have now over 2,500 cases of ovarian cancer processed this way, and 2,000 controls, as well as women who come to the clinic with a suspected ovarian cancer, some sort of mass, but it is benign. And we call those the surgical controls. The collection of these cases and controls, because Mayo Clinic is famous, people come from around the country. Well, of course, we need to have a very good study design, and this is what epidemiologists do. So our approach has been to have a good study design and then focus on annotating the samples with much information. So in our case control comparisons, we limit to women who are living in the area surrounding Mayo Clinic so that we don't include more of the referral, because they may differ by income or disease severity. And those 6 states are shown there. We also limit our case control comparisons to incident cases, so women diagnosed within 1 year, women enrolled within 1 year of diagnosis. The controls we match by specific geographic region as well as age at diagnosis. And so we have a more limited set to make our case control comparison. But also we study in my work the cases who aren't in this collection, that are those that are from outside, those that are more of a prevalent case, because if we're studying survival and clinical questions, they can contribute to that, although not as much for risk. So the epidemiologic principles are important to us so that we can make proper inference, because we are not population-based necessarily. We still are a clinic-based population. So we've done some analyses that we've presented at the Society for Epidemiologic Research to compare the characteristics of women who agree to be in the study versus not agree in the study. Is there a difference between women who provide blood samples and who don't? We compare our characteristics of the cases to true population-based collections that that exists in America called SEER. And then we look at our risk factor frequencies and risk estimates to see if it's as expected. So just to show you one visual of this is kind of a heat map of our distribution of where the women live who are in our case collection and our control collection. And all in all, it does work. The system and our risk estimates match what we would expect, and we monitor it on a monthly basis to make sure that that it still is going okay. So I've introduced the Mayo Clinic and the case control design that we're using, and I'd like to show this slide because it also shows how we have evolved and progressed in this field. So we've been studying classical exposures, cases versus controls. My primary interest has been in the molecular work, looking at genetic variants, which we refer to as SNPs, that are common in the germline DNA. Then we look for inherited risk factors. More so, what I'm going to talk about in this presentation is where the work that we're doing, where we're starting to integrate. So looking at the tumor characteristics and thinking of what are the ways to study tumors in relation to risk factors, in relation to genetic inherited things. And we think that this has good hope for us to better understand risk and outcome in ovarian cancer. This is some pie charts of our distribution of the basic histotypes in our collection, at least in this point in time from back in 2006, but just to remind us that we have started to really feel confident that the different histotypes of ovarian cancer, so high-grade serous, endometrioid, clear cell mucinous, as well as low-grade serous, actually are are distinct diseases. Even though they occur in the same physical location, they're treated the same. I assume here as well that we don't have quite yet, with the exception of PARP inhibitors as a great example, histotype-specific treatment. There's a lot of evidence that comes from the epidemiology, risk factors, as well as molecular factors, and also germline factors. These are different. So in our studies now, we focus on certain histotypes. Again, this is consistent with the studies that suggest that there are different cells of origin for ovarian cancer, and that, for example, high-grade serous ovarian cancer most likely arises from the fallopian tube. So in fact, I think ovarian cancer is a bit of a misnomer, and some people prefer to call it tubo-ovarian cancer. Okay, I would refer you to a fairly recent and very good review article by Brett Reed at Moffitt Cancer Center in Tampa, Florida, who went through the literature and reviewed the molecular and epidemiologic factors that contribute to risk of different histotypes, as well as a review of the different somatic landscape that occurs, for example, BRCA1 and BRCA2 and P53 and high-grade serous ovarian cancer. So there is some variation, even in risk factors for the different histotypes. I'll show you a bit of results now, which is transitioning to thinking about inflammation, the immune system in ovarian cancer. So for this, I'll also introduce the Ovarian Cancer Association Consortium, or we call it OCAC, which is the group where I had the opportunity to meet Francisco. So I feel very grateful, because this has been a very nice friendship. So OCAC is a collection that started, about 15 years ago, for people like myself and you to start to work together to study ovarian cancer, primarily case control collections, but some case series. It now includes over 60 studies. And we primarily have looked at genetic factors. So we require that there is a blood sample available, and we look at the DNA to identify, for the most part, common variation associated with risk, and that was fruitful. We have found over 40 regions in the genome that there is a very believable variant that associates with ovarian cancer risk. But we've also been able to look at epidemiologic risk factors. This is from a recent paper from Britton Trabert at the NCI, National Cancer Institute in the US, who looked at the use of aspirin in OCAC cases and controls, and she was able to show that aspirin use, particularly low-dose regular users, is associated with protection against ovarian cancer. So it's a protective effect and a possible chemo prevention if we get to this point. And it's consistent with the hypothesis that primarily of incessant ovulation. So at the site of ovulation, there's an inflammatory process that leads to some DNA repair issues, and the thought is that that is the site of the origin of ovarian cancer. Not so sure. So I and others thought, okay, let's start to study these tumors in our also across institutions. And so how would we go about this? I would like to look at immune features, but I studied germline, so I'm not sure how. So I've partnered with other pathologists and people who are experts in this field. We had the goal of determining if there are different immune features by tumor histotype. Small studies have suggested it, but on a larger scale, we didn't really know. was also very interested to associate immune features of tumors with these epidemiologic risk factors, such as aspirin use. Does aspirin use contribute to different types based on immune features? And then also we're interested to do molecular subtyping. So some of the decisions that we and others are forced to make is, well, what kind of tissue do you have? What kind of tissue is available? What kind of tissue is ideal? And often it's not the same. So the choices being, should we use formalin-fixed paraffin-embedded tissue that's available in more cases in our collections, which makes it optimal for a sample size. However, the DNA and RNA doesn't always perform as well. And even within the use of paraffin, there are choices to make about, do you use individual blocks of each patient, or should we use tissue microarrays where we core within the tumor tissue and then array it onto within another block, and then you can sample many women's tumors at once for your staining. And this we use for immunohistochemistry. But then also at our institution, but not everybody, we have fresh frozen, and this is preferable because the DNA and the RNA extractions perform very well, and it has very good quality. So that's one of the decisions that one makes. And then for the different types of data, we consider, well, should we look at the proteins and use immunohistochemistry? We can, there's different approaches to doing that. You can look one marker at a time. There are now some multi-marker ways to do immunohistochemistry. If you do that, you're assessing protein, and you can also localize where the expressing cells are. So it ends up, you can get some pretty specific information. On the other hand, if we're interested in RNA, there's technologies for doing that, RNA sequencing. For those studies, we usually just take the tumor and a tissue and say if it's over 70% tumor, and we extract the RNA. So you don't localize within, with the different cells, at least what we've done so far. One of the assays that we've done recently is using this nanostring platform that is a customized RNA expression array, for lack of a better word, that uses paraffin quite well and is inexpensive. I told you about OCAC, which is this case control collaboration. This work also is using OTTA, so Ovarian Tumor Tissue Analysis Consortium, which is set up like OCAC, but it's not restricted to cases controls. It's just cases. Basically, if a collection exists with over 200 cases who have a paraffin-embedded sample and clinical data and survival data, we say, please, yes, let's work together. And this also has been a very nice opportunity to work with the data from Francisco. This group now has over 10,000 tumors collected and studied. We started it about 10 years ago, and there are over 40 active studies. We've done individual IHC markers over the years and published on that. And primarily we use tissue microarrays, so where we just are sampling within each tumor. But you have an economy of scale, really, in assessing. There's pros and cons. We've done some expression studies, so looking at the RNA using that nanostring, like I mentioned, and we're also now starting to do targeted sequencing of extracted DNA from the tumor. So then within that context, this is what we thought through to study immune factors. So we thought we would start with looking at tumor-infiltrating lymphocytes, or TILs, I'll be referring to them. We know from the literature, it's been reported for many years, that TILs associate with improved survival. This is a plot here from the New England Journal of Medicine paper, one of the first, to show that tumors that contain higher levels of lymphocytes inside they do better. Those women have a higher likelihood of survival. So we thought, well, let's make a larger sample size using a standardized approach, because different studies use slightly different cut points for counting their TILs, for example, maybe a different antibody. We wanted to increase the precision of our hazard ratios, our risk estimates. We wanted to be able to look at histotypes, because as I showed, that's important, we think, for the distinct biology. And then the opportunity to see if there's any sort of dose response. So we did staining for CD8 and worked with a pathologist called Martin Coble in Calgary, Canada, who agreed to do all of the scoring. And in this, he defined the TILs as those that are strictly within the epithelial component of the tumor, so within the tumor islets, and he didn't count or look for CD8 cells in the stroma or sort of nearby. This is a bit of detail of just how people work together around the world in this sort of thing. So we have the collaborating studies around the world who did the hard work in obtaining the samples and arraying them onto tissue microarrays. We asked them to provide one section of the tissue microarray that sent it to Mayo Clinic, including Francisco. And at Mayo Clinic, we did the staining for CD8. It's actually a very easy and very, very commonly used antibody looking at CD8. But anyway, we did that centralized in one lab, so the standardized, and then we send the slides that were stained to Martin, shown there on the upper right, who did all the scoring with we don't use image analysis. He prefers just to look through the microscope with his eyeball. So he did the scoring and then sent the scores to the central coordinator, Susan Ramus in Australia, who also had received the clinical features of each case from all the studies, and makes the nice data set that then they send back to Mayo Clinic for our data analysts to start to analyze. So it really does take quite a lot of coordination, but we have done it successfully, and it works. This is a picture of representative photos of the different categories that Martin used to classify cases into those that have no CD8 positive TILs, and then other thresholds based on the number of TILs that he could see within a certain field. So I showed you I talked to you about how the data flowed. Once we got the data, this is what we did. So we combined the value across multiple cores per case. Most of the tumors are in triplicate on a tissue microarray, so we used to use the maximum. We looked at overall survival, so time from diagnosis to death, accounting for delayed study enrollment or left truncation, and censoring at 10 years, conducting histotype-specific analysis adjusted for age, stage, and study site, and we did some subset analyses as well. And this is what we published in JAMA Oncology 2017, where with a large sample size, having over 3,000 high-grade serous cases, for example, we were able to show a dose response. There's a trend. So the more CD8 positive TILs you have, the better you do. We knew this on another level, but now we know at a deeper level. And statistically, it is consistent with the dose response, so not necessarily a threshold effect. Apologies if this is a bit hard to see, but we also, in histotype-specific analysis, could estimate the risk per TIL category, and we do have highly significant association within high-grade serous. We're also able to report the endometrioid significant at 0.05, as well as mucinous, and not so sure about clear cell, and not so sure about low-grade serous, because we still have small numbers with those being rarer. within high-grade serous ovarian cancer, we also wondered, does the association of CD8 positive TILs and survival differ by extent of residual disease, by known BRCA1 or 2 mutation, or by whether or not the patient had first-line chemotherapy? And we did not see any evidence of an interaction. It looks like this is a consistent association in every group, with a possible exception of BRCA2 mutation carriers, where we don't see this CD8 protective association, but maybe it's small sample size. Because remember, we're dividing the data into many pieces when we explore these hypotheses. So the next steps for this is we have more work underway using multicolor immunohistochemistry and also this nanostring digital spatial profiling approach that's very new. We're trying to look at other immune cells in these in ovarian cancers. So, for example, the role of plasma and B cells in the tumor spaces, tumor-associated macrophages, and then Tregs, regulatory T cells. So that work is important and underway. And we're also pursuing the concept of the phenotype of CD8 positive TILs as being of interest in itself. We know it's clinically relevant. Now we want to understand what are the factors that underlie why some tumors are hot, or sort of immunoresponsive, and some tumors aren't. So now we're linking the two consortia data, the OCAC data and the OTTA data, and we've started to do this with a smaller set of studies, 12 studies so far, and we're combining the epidemiologic risk factor data, such as the aspirin data I showed, with the CD8 positive TIL data to see if there's a difference in risk factors among high-grade serous if you are CD8 high versus CD8 low. So analogous to what I think of in breast cancer, ER positive, ER negative. So we have CD8, high versus low, and let's understand the etiology of that, because we know that histology has been important overall. So this immune stuff within histology. We're also linking the common genetic variation that I mentioned with the CD8 positive TIL levels, as well as looking at some of the known mutations. To show you some preliminary data, and just as an example of the kind of approach that works to tackle this, we have started to link the data from our genome-wide association studies, where we looked at and imputed up to 12 million common variants in cases and controls using the Illumina OncoArray B-chip. And here we look within cases only, and so we're looking for a variant. Are there any variants that associate with CD8 positive TILs? That is our endpoint. That's our phenotype among cases. So we generate odds ratios and their relevant p-values and statistics, and we can plot this out on a genome-wide scale and then zero in to see if there is a variant that meets a stringent threshold for statistical significance using ordinal logistic regression. And also we're doing approaches where we can combine across multiple variants using a polygenic risk score. We're also looking to see if within the 40 known regions for overall ovarian cancer, if risks differ by this immune phenotype. Another thing that we're doing now is to ask the question about are there rare variants that relate to CD8 positive TILs? I've showed you some data from our publication that shows that there may be a difference with a woman that has a BRCA2 germline mutation. So we want to make that much deeper and also look at many other genes. So we're primarily working with Fergus Couch at Mayo Clinic. We're applying a targeted sequencing approach for something like 50 DNA repair genes, and looking at the germline. So we have RAD51, we have PALB2, just many different genes. But the analysis is the same, an ordinal logistic regression approach, but here doing more of a burden test, so gene level analysis, and just with some larger sample size of our preliminary data, we see that patterns are holding up what we saw originally. But you can see that we need, to do these analysis, we need larger sample sizes. So we continue to seek funds. So in addition to looking at the IHC, looking, for example, at CD8, we've done some of the RNA-based analyses. Primarily, as I mentioned, this is with the nanostring because it's a cost-efficient way of assessing expression. We've designed with others a panel of 500 genes that are basically candidates that we wanted to look at in over 3,000 high-grade serous ovarian cancers in this collection called OTTA. One of the primary goals was to associate individual gene expression with outcome, so sort of candidate prognostic genes, as well as we wanted to use this panel to classify each tumor into a TCGA sort of derived molecular subtype. So the TCGA has, and others, this Tuttle paper et al., has basically shown that there are molecular subtypes based on RNA expression. There's a mesenchymal and immunoreactive, differentiated, and proliferative among high-grade serous ovarian cancer. So we wanted to basically classify the cases that are in OTTA. And that has since been done. There's a couple of different nanostring experiments, and Francisco is participating in the current one. One of the things, as an aside, that we've learned is that the sampling site within the body matters, that the omental samples tend to be more of a mesenchymal subtype than the non-omental samples, and so that's something that we're taking into account in our analyses. In addition, the assignment into subtype is mathematical, and it does have error, and it is probability-based. And so we're doing analyses where we exclude the cases that seem, they might be mesenchymal, they might be immunoreactive, and so we do some subset analysis or sensitivity analyses to those that really seem to be pure. I mean, issues of tumor heterogeneity are also a huge concern. So with these data, we are now asking these questions. So molecular subtype, one of them is immunoreactive. For example, just from the name, we want to see how that correlates with the extent of CD8 positive TILs. We can use simple statistics to answer that question and test it statistically. The second question that we ask is, well, are these two things, molecular subtypes and the CD8 TIL counts, are they independent? So if we put them into a model, which one wins out, essentially, if we do a Cox regression and adjust one for the other. So we're starting to explore how these relate. And this is some of our early data. This is histograms of looking at the membership of each tumor in the molecular subtype, TCGA based, so mesenchymal, immunoreactive, by its category of CD8 positive TILs. And we see some very, in some ways not surprising results, but in some ways surprising. So immunoreactive classified tumors have higher levels of TILs. Okay, we can show this statistically, we can show this with our data. And so I think, well, yes, of course. But on the other hand, it's quite remarkable because this is despite the fact that one test is with protein, one test is with RNA. Still holds. Despite the fact that the CD8 uses tissue microarrays, it's just a small sampling. You have heterogeneity issues versus the RNA is from a whole block that's just limited to 70% tumor and extracted, ground up. It still is, despite that. And then the fact that the CD8 is an eyeball test to localize specifically where, and that's not what the RNA is. So it's reassuring that we see this. It makes us think, okay, this might be, the data might not all be noise. When we estimate the hazard ratios for CD8 positive TILs and death in ovarian cancer. This is where we expect to see this trend. When we adjust for molecular subtype, we still see this trend, suggesting that there could be some independence. When we do analyses stratified, so where we limit analysis to particular subtypes and see is there a trend within each subtype, not adjusting. The mesenchymal subtype is maybe a bit different. And the mesenchymal if you have a mesenchymal tumor, we don't see a trend of levels of CD8 positive TILs. It may be more of a threshold effect there. You really need to have a lot of TILs in order to have it have an effect on survival. This is what the data suggest. Interesting. So it's generating hypotheses for us to test now in the future. So we're collecting more cases where it's easier to generate more CD8 data. We have a lot already of the molecular subtype and genetic data. So we're filling in the gaps in data availability. And these are some of the challenges that we face in this work. So to harmonize the data across studies around the world is very, very challenging and actually much harder than the molecular data in many respects. The clinical data and the risk factor data needs to go through this harmonization process. It's critical. We have thought through and discussed which is the optimal study design, study material. There's still not one that's obvious. There's advantages and disadvantages to all of the options. And in fact, it's good to have options. We have realized that for us, the multicolor immunohistochemistry is actually much more difficult to score than I expected. But it's quite difficult, and it's a hurdle to do this with a computer or with an eyeball. When we do our analyses, we also still are quite simple. Everything I showed you is basic data analysis 101. And so there's many, many more things that we could do. We start with the simplest and see what answers we get. So there's a lot of challenges in studying immune factors with an epidemiologic framework. So some ideas for the novel approaches is what we're quite excited about. And it's, again, because the immune phenotypes are quite largely predictive of outcome, and this is in numerous traits. It's not just ovarian cancer. Unfortunately for ovarian cancer, the immunotherapies have not worked largely. So while we have hope in other cancers, it's a real problem in ovarian cancer. And so it's possible that if we could stratify tumors in patients into these immune-related groups and understand more about the biology, we could find a better targeted population in which to do the immunotherapy trials. And actually that is underway in Australia, some subtype-specific trials. We've also learned that we really need to bring in the pathologists early, and we're very, very grateful, and have like diverse teams. Again, using large sample size, because subset analyses, the power gets reduced quite a bit. And then it's a real strength and a key to have a set of samples that has multiple data types. So then you can compare epidemiology, genetics, protein, et cetera. And then so we really need to have some additional computational approaches. So very glad that bioinformaticists exist. So I'm going to close and share with you some thoughts. So going from Dr. Mayo to Professor Francisco. This has been a fantastic visit and journey, and I really appreciate what I've been learning here. The experiences of meeting people from many disciplines who have outstanding training and experience has been very inspiring. I encourage to have very nice, large, properly executed biobanking, biorepositories. It's very exciting that this is happening here. I believe real data from our own patients is what is best to derive novel bioinformatics and analytical approaches. I have spent time developing new approaches And that's fine. I like to do this, but it's based on, we need to make it based on our own patients and our own experiences. And so that's also something that I encourage and emphasize usually. And it's also critical, like I've seen at Mayo, the investment in early infrastructure, it doesn't pay off immediately, but it does pay off. And the years go by quite quickly, and then before you know it, you have amazing resources. So I'm really find that that's important in epidemiology. And then it's very good to act within your own institution, but also to collaborate, as I have done. And I just want to thank some of my colleagues at Mayo Clinic and beyond who contributed to this work and the creation of all of these data. So with that, I'm happy to take questions about anything that I presented. Thank you very much.